Hello dear students, this is Dr. Devyani Sharma from the Faculty of Science, Sri Rawatpura Sarkar University, Raipur, Chhattisgarh. In this video lecture, we will be discussing about MAP kinases. So let us start today's lecture. MAP kinases stands for mitogen activated protein kinases. These are the proteins that are serine threonine specific kinases which are activated by a wide range of stimuli including pro-inflammatory cytokines, growth factors, mitogens, osmotic stress, heat shock and directing cellular responses. So basically these MAP kinases are intermediates in the signal transduction pathways that are initiated by many types of surface receptors. And the targets for MAP kinases are located within many cellular compartments. And they act as a physical link in the signal transduction pathway from the cytoplasm to the nucleus. The first ever mitogen activated protein kinase to be discovered was ERK1, also called as MAPK3. Function in a signaling cascade are activated upon ligand binding to a cell surface receptor, activating several MAP ER kinases, which in turn phosphorylate their respective substrates. So each MAPK pathway contains several layered or tiered kinase cascade. They comprises of various MAP kinases. So these cascading events regulate various cellular activities such as the gene expression, mitosis, differentiation, proliferation and cell survival or apoptosis. These MAP kinases are found in the eukaryotes only, but they are fairly diverse and encountered in all the animals, fungi and plants, and even in an array of unicellular eukaryotes. The closest relative of MAPKs are the cyclin-dependent kinases, which are an important part in the cell cycle. So upon the extracellular mitogen binding to the ligand, RAS, that is RAS, which is a GTPase, exchanges GDP for GTP. So this in turn initializes a cascade activating MAP3K, that is RAF, which in turn activates the MAP2K and which activate the MAPK. So this is a cascading activity where the MAPK can activate a number of transcription factors which control key processes in the cell. For example, epidermal growth factor, that is EGF, binds the EGFR, which activates the tyrosine kinase activity as shown in the figure. So GRB2 and SH2 domain containing the proteins, they recognizes the phosphorylated residues along with a guanine nucleotide. Then binding to the EGFR takes place. Here again the GDP is then swapped for GTP, which binds RAS to make it active. So this is basically a cascading process which is explained in the particular figure here. And this particular RAS, which is formed at the end of the previous cycle, it activates the RAF, which can phosphorylate and activate another MAP kinases called as MEK1 and MEK2, which in turn activate the MAPK. So MAPK, which is the main regulating protein here, it regulates a number of transcription factors like CMYC, CREB and CFOS, etc. So all these regulate the expression of a number of genes involved in the apoptosis, differentiation, cancer, etc. So defects in the regulation of these pathway can lead to the uncontrolled growth in the cells, which leads to cancer. And in humans, there are at least 11 members of the MAPK family, which can be divided into several subgroups also. And let us discuss about the activation of MAP kinases. So basically the mitogen activated protein kinases are catalytically inactive in their base forms. In order to become active, they require phosphorylation event in the activation loops, as it can be seen in the figure, which is an X-ray structure of the ERK2 MAP kinase in its active form. So phosphorylated residues are displayed in red. So over the last decade, there are some important advances which have taken place in developing novel agents that modulate key proteins in this pathway, culminating in the approval by the FDA. For many years to come. So this was all about MAP kinases. For any query, please put on your comment in the comment section, and do like and share the video, as well as subscribe to the channel. Thank you for now.